Today we'll be looking at how antithrombin as a protein acts in the body and where it comes from. Antithrombin is made from DNA. The four basic subunits of DNA are called nucleotides. These nucleotides can be seen here: A, T, C, and G. These nucleotides pair in a specific way. A can only bind with T and C can only bind with G. In this way, there are plenty of combinations that can be made in the DNA which form proteins. DNA is read by parts of the body, by processes in the body, which create proteins. The protein created here from DNA is antithrombin. We can see antithrombin here as a dimeric molecule, meaning that there are two parts to this protein, or two chains. In the middle of this protein is a reactive center loop. This is where the function of antithrombin comes from. This reactive center loop, when activated, pops out of the molecule and cleaves thrombin, preventing its action and stopping clots from forming in the body. However, antithrombin is helped along by heparin, another protein. Heparin binds to specific heparin binding sites on the I chain of antithrombin. We're now going to have a look at how antithrombin works in the body. Thrombin acts on fibrinogen by breaking fibrinogen down into insoluble fibrin units. These insoluble fibrin units band together and form a net, or like a clot, as we can see here. This prevents the blood from flowing through. This process is extremely useful if we get cuts or injure ourselves and need to stop blood flow. However, if it is left unchecked, it will eventually cut off blood supply. So this is where the mechanism of antithrombin comes in. Heparin binds to specific binding sites, 1, 2, 3 for example, but there are many more, on the antithrombin molecule. When heparin binds, it produces a conformational change which changes the shape of antithrombin. This shape pushes the dimeric molecule into a different configuration. What this allows is the reactive center loop to come out of the middle of the protein. Now we can see the reactive center loop in the middle of antithrombin here. This is what will cleave thrombin and act and create antithrombin's primary action. It is important to note, however, that antithrombin can still work in this inactive form, as we like to call it. However, it is not as efficient. The binding of heparin is what creates an efficient and properly working antithrombin protein. From here, this is an activated antithrombin. This antithrombin is now exposed with the reactive center loop. And this reactive center loop can perform its cleavage function on thrombin. As we can see here, thrombin fits perfectly into the reactive center loop pocket, thereby creating a specific fit to ensure that antithrombin doesn't cleave other molecules that are not thrombin. In this way, antithrombin is a specific protein. The resultant of this cleavage creates a broken down or inactive form of thrombin, which prevents it from performing its previous function. By doing this, we can now see that the action of thrombin can be inhibited. If thrombin can't act on the insoluble fibrinogen, then we can't get this soluble net of fibrin forming together down in the blood clot. And now here is an example here. Now that thrombin cannot perform this pathway, fibrinogen stays in the body as an inactive unit and allows blood to keep flowing along. This ensures that blood supply is maintained. We're now going to look at a mutation which occurs in antithrombin, which occurs in some individuals' DNA, resulting in a defective antithrombin protein. When we looked at before how A can only bind with T and C can only bind with G, this is still true. However, what happens when this process of DNA goes wrong? On the exon 6, which is a coding region or a region of DNA which codes for protein of the antithrombin gene, there can be a mutation of a G nucleotide to a T nucleotide at this specific location here. When this occurs, we get the resultant amino acid, which is read by the body, or translated as we call it, results in a different amino acid. So when we talk back about our primary structure in the protein structure part of the website, there's information on how amino acids are formed and what a polypeptide is. Now that we know this, when an amino acid changes from, for example here, a serine to an alanine at this specific residue site, we get a different shape of antithrombin protein. We can see here that clearly there is a different shape of antithrombin in relation to the active site, the reactive center loop in the middle. This stalk, which connects the reactive center loop and part of the antithrombin dimer, the L chain, is misfigured in this form of protein. We call this a mutation. More specifically, we call this a substitution mutation. Here is the molecule, the mutant molecule, preparing for binding with heparin. As heparin binds, we usually would expect to see both dimeric sides, both dimers of the antithrombin, expanding out and revealing our reactive center loop so it can perform its function. However, in this mutant, we get movement in the I chain, but not movement in the L chain. And this prevention of movement means that this reactive center loop here is partially expelled, but not over here. This is going to prove to be a problem. Now, we see when thrombin here, the thrombin molecule comes in to bind, because antithrombin is looking to perform its function, which is cleaving thrombin and preventing that blood clot pathway, there is an imperfect fit here. It binds well on this left side, we've got contact here, and so that's binding well. However, this right side here, there is not proper binding. Now, an important distinction to make here is that it will still act on thrombin, but not as efficiently. So this mutant antithrombin molecule will cleave some thrombin, but it will not result in proper efficient mechanism that we need in the body. This can be seen here. So when this mutant antithrombin acts on thrombin, we get both some cleaved thrombin occurring, which is good, but then some that is not cleaved, which is bad, which essentially means that we are not actually stopping this pathway from occurring. So then fibrinogen is still broken down to fibrin, and fibrin still forms this clotting net that we saw before. And this has been seen to lead to recurring cases of deep vein thrombosis, or DVT. As a consequence, individuals can suffer from recurrent episodes of DVT, which can be both life-threatening, limb-threatening, and is a serious disease that we need to look at.